Based on the physiological experiments of other labs, in particular the heart rate experiments performed at IDIBAPS, we use this internal variable as a correlate of one of the subsystems driving the artificial rate. This line is the plot on real time of the arousal felt by the robot. When we see the arena, color of red indicates a maximum arousal. So here we see two subsystems interacting. In green, we have the security in time, and for example, in this area, the rat stayed longer in the home base. And in red, we have the fear to open space, and each peaks would correspond to traversals in the rat trajectory of the arena. In the right, we have the same plots, but now generated by the artificial model. So on top of these two subsystems, we can add a third one, considering the motivation of the robot towards obtaining reward. In this plot indicated by these blue cues. We are still staying in a reactive level, so an immediate response is generated from the integration of the different subsystems. This model driven by self-regulation would just be a first building block of a more complex cognitive architecture. On top of this, we could consider adding learning capabilities and other layers to what would be a more complex cognitive architecture driving this artificial agent.
When we forage for something. What we want to know as a first pass is what's happening in the rodent brain as it is foraging.
This is a view from the rat brain atlas, where lots of tiny areas are distinguished based on cell characteristics. Here we find in a staining the neocortex, the piece right below the skull, and below we have the hippocampus over here. The width of the hippocampus is about 4 millimeters. And we place our electrodes in a tiny cell layer over here that contains many cells, and what we then hope to record is lots of action potential signals, spikes or impulses, as well as more continuous slow fluctuations in mass activity.
So here you can see the layout of the drive. We have 14 recording tetrodes which are in a circle. And we are aiming for the CA1. It's just 14 microns thick, so we have to be very careful lowering the tetrodes down. We will slowly move them down each day until we reach the hippocampus. Each line is the input you get from one of the tetrodes. We're now listening to the spike sounds of tetrode 10, which you can see here. By listening and by hearing the different patterns of spiking, you can actually differentiate between different types of cells, and you can know where you are. So are we layer 6 of the cortex? Are we in the hippocampus? You can see your cellular activity, but you can also hear. Very specific of the hippocampus are the sharp waves, which are the sounds. The sharp waves usually point towards the layer, so it's a nice indication if we have to move up or down, if we're above the layer or below the layer. And the bigger the ripple, the more we are actually in the layer. So another specific thing about the hippocampus is you can see the theta rhythm. This tetrode is slightly too deep, so it's going more towards the fissure. There you have a lot of big theta activity.
Now, theta activity is a significant, one of the most significant, maybe the most significant oscillatory pattern which modulates the activity of cells in the hippocampus. The errors introduce rats into a maze, not very big, still kind of small maze. In this maze, when rats are introduced to the maze, they pick one of the corners as their home base. Following that, when they are already adapted to this home base, they are starting exploration. They move outside of the home base, they explore the maze, and they come back to their home base. Our main interest is to see how hippocampus is marking the space by emotion. Not only by time, but also it's marked by by emotional attitude we have to certain zones of the space or certain objects in the space. In one of these experiments, Erez introduced a small pellet of food in a distant corner of the maze. The animal learned that whenever he is going now outside of the home base, exploring the maze, reaching this corner, he finds this small food pellet and it consumes it. In further experiments, Erez introduced a large food pellet in the same corner. Now the animals learn that when they reach this corner, they find a large piece of food. This big chunk of food is not consumed on the spot, but rather is carried back to the home base, and what we call hoarding behavior. Under these conditions, rats showed increase of theta frequency on the way from the home base to the corner with a large pellet of food, and also on the way back to the home base where they are consuming the food. So now the conclusion from these two experiments is the following. We see that theta frequency reflects expectation of some reward, positive reward. It is either finding the food and consuming it on the spot, or finding the food and then carrying it that, and consuming it later on back in the home base. Theta is affected by expectancy of reward. This implies the possibility that actually expectation to find in some place in the space, in a known place in the space, some significant reward may affect the mapping of the space by the hippocampus.